Now, let's start learning about DNA replication. Transmission of chromosomal DNA from generation to generation is crucial to cell propagation. This can only be achieved when chromosomal DNA is accurately replicated, providing two copies of the entire genome for faithful distribution into each data cell. Semi-conservative replication. It is crucial that the genetic material is reproduced accurately. When Watson and Crick worked out the double helix structure of DNA in 1953, they recognized that the complementary nature of the two strands, A paired with T and G paired with C, might play an important role in its replication. Because the two polynucleotide strands are joined only by hydrogen bonds, they are able to separate without requiring breakage of covalent bonds. If the two strands of a parental double helix of DNA separated, the base sequence of each parental strand could serve as a template for the synthesis of new complementary strand, producing two identical progeny double helixes. This process is called semi-conservative replication because the partial parental double helix is half conserved, each parental single strand remaining intact. The alternative methods are conservative and dispersive. In conservative replication, in which the whole original double helix acts as a template for a new one daughter molecule would consist of the original parental DNA and the other daughter would be totally new DNA. In dispersive replication, some parts of original double helix are conserved and some parts are not. Daughter molecules would exist of part template and part newly synthesized DNA. So Michelson and Stahl experimentally demonstrated the semi-conservative replication of DNA in E. coli in 1958. Other methods include conservative and dispersive. Okay. Conservative and dispersive. The figure illustrates a prediction of this model. If the parental DNA carries a heavy density label because the organism has been grown in medium containing a suitable isotope such as 15N, its strands can be distinguished from those that are synthesized when the organism is transferred to a media containing normal light isotopes. So generation 1, cells grown in 15N, then replication. So after centrifugation, so generation 1, cells grown in 15N, then N15, replication after centrifugation. Then generation 2, cells grown in 14N, then hybrid is formed. Generation 3, cells grown in 14N, light and hybrid. The Michelson-Stahl experiment showed that DNA replicates by a semi-conservative mechanism. In this experiment, E. coli cells initially were grown in a medium containing ammonium salts prepared with heavy nitrogen until all the cellular DNA was labeled. After the cells were transferred to a medium containing the normal light isotope, samples were removed periodically from the cultures and the DNA in each sample was analyzed by equilibrium density gradient centrifugation. This technique can separate heavy, light, and heavy light duplexes into distinct bands. After one generation of growth, all the expected DNA had the density of 14 and 15 N DNA. After two generations, approximately half the DNA had the density of 14 N and 15 N DNA. The other half had the density of 14 and 14 N DNA. With additional generations, a larger and larger fraction of the extracted DNA considered the 14N duplexes complexes never appeared. This results match the predicted pattern of the semi-conservative replication mechanism. Then replication and origin of replication. DNA replication does not start at random locations but at particular sites called origins of DNA replication. A DNA whose replication starts from an origin and proceeds bidirectionally or unidirectionally to terminal sites is called a replicon. The unit of DNA replication for karyotic replicons are usually circular. In bacterial cells, the circular chromosome contain a unique origin and DNA replication process bidirectionally from the origin to the terminus. Therefore, a whole bacterial genome is a single replicon, monorepliconic. On the other hand, eukaryotic cells contain multiple replications origins on each chromosome and hence they many replicons, multirepliconic. Individual replicons in eukaryotic genomes are relatively small and generally 40 to 100 kb in size. The E. coli origin of replication is referred to as ORIC. This spans approximately 249 base pairs of DNA. It contains two short repeat motif, one of 9 nucleotides and the other of 30 nucleotides. The 9 nucleotide repeat, 5 copies of which are dispersed throughout ORIC, is the binding site for a protein called DNA A. The result of DNA A binding is that the double helix opens up or melts within the tandem array of 380 rich 13 nucleotide repeats located at one end of the ORIC sequence. Here we could see E. coli origin of replication. ORIC contain repetitive 9 BP and 80 rich 13 BP sequences referred to as 9 mers and 13 mers respectively. So multiple copies of DNA A protein bind to the 9 mer and then melt to the 13 mer segments. 9 mers, then 13 mers, ATP plus DNA A, initial complex and opened complex, supercoiled template. Initiation at ORIC occurs after DNA A protein binds the 5 9 mers. The 13 mer region is then denatured, and this open complex serves as a replication site adapted. And into that, then ORIC contains 11 5' GATC 3' repeats that are methylated on addition on both strands. Methylated on adenine on both strands. Only fully methylated origins can initiate replication. Hemimethylated daughter origins cannot be used again until they have been restored to the fully methylated state. Origins of replication in yeast is known as autonomously replicating sequences, or ARS. A typical yeast ARS is shorter than the E. coli origin, usually less than 200 base pairs in length. As ARS element consists of an AT-rich region that include a consensus sequence, A domain, and additional elements. A domain contains 14 base pair sequence and that contains 11 base pair consensus sequence consisting of 80 base pairs. In metazoans, no clearly conserved sequence elements have been identified at replication origins. And common features of replication origins. Replication origins are unique DNA segments that contain multiple short segmented sequences. These short repeat units are recognized by multimeric origin binding proteins. These proteins play a key role in assembling DNA polymerase and other replication enzymes at the site where replication begins. Origin regions usually contain an AT-rich stretch. This property facilitates unwinding of double duplex helix because less energy is required to melt AT base pairs than GT base pairs. Then replication fork. In each replicant, replication is a continuous form and process, and the form of the origin to the terminus and is accomplished by the movement of the replicating point called the replication fork. In case of unidirectional replication, single replication fork forms, whereas in bidirectional replication, two replication forks form. Most DNA replication is bidirectional. So origin of replication is there. Replication fork is formed, direction, unidirectional replication. But in bidirectional replication, origin of replication fork, direction, leading, lagging, replication fork, and direction. Then unidirectional and bidirectional replication. The point at which replication is occurring is called the replication fork, sometimes also known as growing point. A replication fork moves subsequently along the DNA from its starting point at the origin. In bidirectional replication, two replication forks are formed. They proceed away from the origin in opposite directions. The velocity of fork movement can be estimated from the size of replicant and the length of its replication period. In E. coli, the replication fork proceeds at about 1000 base pair per second per fork, and one Okazaki fragment is synthesized every 1 to 2 seconds. In eukaryotic cells, the rate of fork movement is slow. So unidirectional and bidirectional replication, the point at which replication is occurring is called the replication fork, also known as the growing point. A replication fork moves sequentially along the DNA from its starting point at the origin. In bidirectional replication, two replication forks are formed. They proceed away from the origin in opposite directions. So DNA replication in E. coli. The genome of E. coli is replicated bidirectionally from a single origin, ORIC. E. coli replicon is circular with no free ends. Replication of DNA in E. coli is also known as theta replication, and it occurs in 3 steps. Initiation involves recognition of the position on DNA molecule where replication will begin. Elongation concerns the events occurring at the replication fork. where the parent polynucleotides are copied. Termination occurs when the parent molecule has been completely replicated. Biochemistry of RNA replication. DNA replication. DNA replication is a complex process involving numerous enzymes at the replication fork. In E. coli, more than 20 different proteins participate in DNA replication on the basis of their biochemical roles in different stages of chromosomal DNA replication. These proteins are classified into several categories, like proteins involved in the initiation of replication: DNA A, DNA gyrase, single-stranded DNA binding protein SSB, DNA B, helicase, and DNA C. DNA A, DNA B or helicase, then DNA C. Then proteins required for chain elongation and connecting Okazaki fragments: DNA G primase, DNA polymerase, SSB, and DNA ligase. A swivel to relieve torsional stress in advance of the replication fork, DNA gyrase. Proteins for the termination of replication and segregation of daughter molecules, terminus binding protein, and the DNA topoisomerase 4. Then DNA helicase and primase. DNA primers require single-stranded DNA as a template. Cannot synthesize a chain without an RNA or DNA primer annealed to the template. Two other enzymes enable polymerase to work on a double-X DNA. One is a DNA helicase, which opens up the duplex at the replication fork to provide a single-stranded template. The primary replicator helicase, or DNA B, bind to and move on the lagging strand template in the 5' 3' direction, unwinding the duplex as it goes. This helicase action requires ATP hydrolysis. The separated strands are inhibited from subsequently re-annealing by single-stranded binding protein, or SSB protein, which binds to both separated strands. The other is a primase, which synthesizes a short RNA primer to primer DNA chain elongation. The term primosome is used to denote a complex between primase and helicase, sometimes with other accessory proteins. Then topoisomerase. A DNA topoisomerase can be viewed as a reversible nuclease that adds itself covalently to a DNA backbone phosphate, thereby breaking a phosphodiester bond on a DNA strand. Once again, a DNA topoisomerase can be viewed as a reversible nuclease that adds itself covalently to a DNA backbone phosphate, thereby breaking a phosphodiester bond in a DNA strand. This reaction is reversible and the phosphodiester bond reforms as the protein leaves. One type of topoisomerase, called type 1 topoisomerase, produces a transient single-stranded break. A DNA topoisomerase can be viewed as a reversible nuclease that adds itself covalently to a DNA backbone phosphate, thereby breaking a phosphodiester bond in a DNA strand. This reaction is reversible and the phosphodiester bond reforms as the protein leaves. One type of topoisomerase, called type 1 topoisomerase, produces a transient single-strand break. This break in the phosphodiester backbone allows the two sections of DNA helix on either side of the nick to rotate freely relative to each other. Using the phosphodiester bond in the DNA strand opposite the nick as a swell point, any tension at the DNA helix will drive this rotation in the direction that relieves the tension. Because the covalent linkage that joins the DNA topoisomerase protein to a DNA phosphate retains the energy of the cleaved phosphodiester bond, revealing a rapid and does not require additional energy input. Topo 1 from E. coli acts on negative but not positive supercoils. A second type of DNA topoisomerase, type 2 topoisomerase, forms a covalent linkage to both strands of the DNA helix at the same time, making a transient double-stranded break in the helix. The first type 2 topoisomerase, topo 1, 2 to be described was isolated from E. coli and named DNA gyrase. In prokaryotes, topo 2 changes the positive supercoil into a negative supercoil of the increasing number of negative supercoils by 2. These enzymes are activated by sites on chromosomes where two double helices cross over each other. Once a topoisomerase 2 molecules bind to such a crossing site, the protein uses ATP hydrolysis to perform the following set of reactions effectively. It breaks one double helix reversibly to create a DNA gate. It causes the second nearby double helix to pass through this break, and it then reveals the break and dissociates from the DNA. All type 2 topoisomerase also catalyze catenation and decatenation, that is, the linking and unlinking of two different DNA duplexes. nalidixic acid and ciprofloxacin and novobiosin binds to E. coli DNA gyrase and inhibits its action and hence DNA replication. Novobiosin blocks the binding of ATP to gyrase. Nalidixic acid and ciprofloxacin, in contrast, interfere with the breakage and rejoining of DNA chains. DNA with two supercoils, type 1, one stranded broken, then both strands broken. One strand passed through and released, both strands passed through each other and resealed. Earlier is also resealed. Action of the two types of topoisomerase. The type 1 topoisomerase break one strand of DNA and pass the other strand through the break, removing one supercoil at a time. The type 2 topoisomerase break both strands and pass another part of the same DNA through the breaks, introducing or removing two supercoils at a time. Some viruses like SV40 are closed circular DNA carrying nucleosomes. If the SV40 virus is treated with topoisomerase and the histone is then removed, it is found to still be supercoiled. However, if histones are removed before topoisomerase treatment, the DNA is relaxed. The DNA makes 1.75 left-hand superhelical turns about each nucleosome. Thus, turns are constrained and cannot be removed while the histones are still present. However, if the histones are first removed, the DNA so produced will have unconstricted supercoils which can be relaxed by topoisomerase. Then DNA polymerase, in an enzyme that can synthesize a new DNA strand on a template strand is called DNA polymerase. Both prokaryotic and eukaryotic cells contain multiple DNA polymerase activity. Only some of these enzymes actually undertake replication. Sometimes they are called DNA replicases. The others are involved in subsidiary roles in replication and or participate in repair synthesis. There are three major enzymes that will polymerase nucleotides into a growing strand of DNA. In E. coli, these enzymes are DNA polymerase 1, 2, and 3. DNA polymerase 1, discovered by Arthur Kornberg, who subsequently won the Nobel Prize for his work, is primarily utilized in filling small DNA segments during replication and repair process. Which one? DNA polymerase 1. DNA polymerase 2 can serve as an alternative repair polymerase. It can also replicate DNA if the template is damaged. DNA polymerase 3 is the primary polymerase during normal DNA replication. DNA pol 4 and DNA polymerase 5 are Y family DNA polymerases and have no proofreading ability. They replicate damaged DNA. In E. coli, pol 3 holoenzyme is the major replicative polymerase for both leading and lagging strand synthesis. The pol 3 holoenzyme is a huge multi-protein complex that consists of 10 distinct polypeptides. The hallmarks of pol 3 is very high catalytic potency and possessivity. Possessivity refers to the ability of an enzyme to catalyze many consecutive reactions without releasing its substrate. The enzyme extends the DNA chain with a high possessivity. And a high catalytic efficiency. Velocity of chain elongation is about 1000 nucleotides per second at 37 degrees Celsius. This replication complex is an asymmetric dimer. The catalytic core of DNA polymerase 3, composed of alpha theta subunits, contains the polymerase activity and 3' to 5' exonuclease for proofreading. A dimer of beta subunits forms a ring-shaped structure and acts as a sliding clamp. Loading of the beta clamp onto the DNA is facilitated by 5 subunit subassembly called gamma complex. Dimerization component causes two core polymerase molecules at the fork to link together and form dimer. Leading strand synthesis, lagging strand synthesis. Then T subunits maintain dimeric structure. DNA polymerase 3 holoenzyme polymerase involved in replication of bacterial genomes. DNA polymerase 1, 2, and 3. ഒന്നാമത്തെ first DNA polymerase 1, DNA repair and gap filling. DNA polymerase subunit 1, Exonuclease activity 3' to 5', 5' to 3' activity is also there. Function DNA repair and gap filling. DNA polymerase 2, 7 subunits, only 3' to 5' activity. Then DNA repair. DNA polymerase 3, at least 10 subunits. Exonuclease activity 3' to 5', no 5' to 3' activity. Main replicating enzyme. The 5' to 3' exonuclease activity of polymerase 1 is independent of both its 3' and 5' exonuclease and its polymerase activities. Proteases such as subtilsin or trypsin cleavage pole 1 into two fragments, a larger C-terminus or clenov fragment which contains both the polymerase and the 3' 5' exonuclease activities, and a smaller N-terminal fragment which contains the 5' 3' exonuclease activity. In eukaryotic cells, nuclear DNA replication requires 3 polymerase, pole alpha, pole delta and pole eta. Gamma is a sole polymerase participating in mitochondrial DNA replication. Nuclear polymerase beta, a small single polypeptide, functions in base excision repair. These are many other polymerases that were recently identified, but biological roles of these remaining polymerases are unknown. DNA polymerase delta alpha is unusual because it has an ability to initiate a new strand. It is used to initiate both the leading and lagging strands. Among the replicated polymerase alpha is unique in possessing a primase activity, and only such activity so far identified in eukaryotic cells, suggesting that polymerase alpha may play a role in priming of DNA synthesis. However, DNA synthesis of polymerase alpha lacking the 3' 5' exonuclease activity is inaccurate and shows a less processivity. These enzymatic characteristics make polymerase alpha poor candidate for the major replicative polymerase. The polymerase alpha binds to the initiation complex at the origin and synthesizes a short strand consisting of about 10 bases and RNA followed by 20 to 30 bases of DNA. Then it is replaced by an enzyme that will extend the chain. On the leading strand, this is DNA polymerase delta. This event is called polymerase switching. DNA polymerase delta and polymerase both possess a 3' 5' exonuclease activity and are able to carry out highly processive DNA synthesis with the aid of sliding clamp and clamp loader. Therefore, these polymerases are better suited for chromosome replication. DNA polymerase delta elongates the leading strand and polymerase may be involved in lagging strand synthesis. Apidicolin, a tetracyclic diterpenoid, is a potent inhibitor of mammalian nuclear DNA polymerase. It does not affect mitochondrial DNA polymerase. So, DNA polymerase alpha, gamma, delta. Then DNA polymerase epsilon and DNA polymerase K. So, DNA polymerase alpha subunits 4, 3' to 5' activity absent, but then function priming during replication. Then DNA polymerase gamma 2 subunits, 3' to 5' exonuclease activity, mitochondrial DNA replication. Then DNA polymerase delta 2 or 3 subunits, exonuclease activity 3' to 5', main replicator enzyme. Then DNA polymerase epsilon, at least 1 subunit. Then exonuclease activity 3' to 5', requires for detection of damage during genome replication. And DNA polymerase K, kappa particle, or 1 or 2 subunits, requires for attachment of cohesin. So, first process will be the initiation. DNA A protein initiates replication in E. coli at ORIC. DNA A protein binds to the 9 MERS sequences in ORI, forming an initial complex. Binding of DNA A to the ORIC 9 MERS facilitated initial strand separation or melting of E. coli duplex DNA, which occurs at the ORIC 13 MERS. This process requires ATP and forms open complex. Further melting of the 2 strands of E. coli chromosome to generate unpaired template strands is mediated by DNA B protein, a helicase. 1 molecule of DNA B, a hexamer of identical subunits, clamps around Further melting of the two strands of E. coli chromosome to generate unpaired template strands is mediated by DNA B protein, a helicase. One molecule of DNA B, a hexamer of identical subunits, clamps around each of the two single strands in the open complex formed between DNA A and ORIC. This binding requires ATP and DNA C protein. Helicase move along a DNA duplex utilizing the energy of ATP hydrolysis to separate the strands. In E. coli, the separated strands are inhibited from subsequently re-annealing by a single-strand binding protein or SSB protein, which binds to both separated strands. DNA polymerase can only elongate existing primer strands of DNA or RNA. The primers used during DNA replication in both prokaryotes and eukaryotes are short RNA molecules whose synthesis is catalyzed by the RNA polymerase primase. After the bond primase synthesize short primer RNA complementary to both strands of duplex DNA, they dissociate from the single-stranded template. When elongation, DNA polymerase catalyzes step-by-step addition of deoxyribonucleotide units to DNA chain. The chain elongation reaction catalyzed by DNA polymerase is a nucleophilic attack by the 3' hydroxyl group of the primer on the innermost phosphorus atom of the deoxyribonucleoside triphosphate. A phosphodiester bridge forms with the concomitant release of pyrophosphate. The subsequent hydrolysis of pyrophosphate by pyrophosphatase, a ubiquitous enzyme, helps to drive the polymerization forward. Elongation of the DNA chain proceeds in the 5' to 3' direction. Then structure of the DNA chain and mechanism of growth by addition of a deoxyribonucleoside triphosphate at the 3' end of the chain.